class. Welcome to our unit of notes on the introduction to biology, specifically biological principles. Today we're going to focus on what does it mean really to do science, to answer questions using science. What's its purpose and function, and what is the kind of processes that we undergo to answer these questions. So the first thing really to kind of highlight is you've probably learned about it before, called our scientific method. In reality, it's not necessarily one method, but it's a way of thinking by following steps and gathering evidence and forming conclusions based on that evidence. So basically, we're going to create claims, gather evidence, and then reason to a conclusion using this evidence and nothing else. The process is really a cycle, which is important to understand that we really do this process multiple times throughout researching any given topic in a scientific community. So there's our question. We learn a little bit about it so we can be educated as much as possible, and then we construct our hypothesis or the claim. All right, so we're going to form a claim about what we believe to be true or what we believe is going to happen. And then from that claim, we move on to designing experiment or testing. So keep in mind, all starts with a question or noticing our initial observation, and we move through. Test of the experiment. How's everything looking? If it's looking good, and we get some data, that's great. Then we analyze our data, and then we start to move through and say, hey, if the results start to support the hypothesis or do not support the hypothesis, you come all the way back up, form another claim, modify your claim. If they do support the hypothesis, then you can communicate your results and you have something meaningful to present. So moving on to the next overall concept would be identifying what our variables are. So before we really even have a claim, we need to or at least at the same time as a claim, we need to identify our variables of our experiment. What our variables are essentially the items in the experiment that you're dealing with. And what are you dealing with in science? We want to control things as much as possible. We want to have a good independent variable, something you're interested in testing. It's what are you testing? You change it. You're in charge of this one. So we want to have one of these. An example in this picture here is the type of liquid that they use. So that would be an independent variable. A dependent variable, what is it? You measure it. What are you recording? What is the thing you want to see happen? So the change happens in here. So this would be the height of the plant. So they're going to figure out how does the liquid affect the height. So independent, dependent. I'll abbreviate these. Important to note: IV, DV, independent, dependent variable. The final thing, the constant or controlled variables. I'll just usually refer to them as controlled variables in class. These are conditions that we keep the same. Everything we want constant and unchanging. So if we're doing this experiment, we should probably have the same type of plant we're testing if we're going to compare how the liquid impacts that. Comparing two different kinds of plants doesn't make a whole lot of sense. We want to use the same type of pot, same size pot to plant them in, same amounts of liquid, so they're getting equal amounts but different liquids. So hopefully that clears it up as to we want to have good controlled experiments in our design, and that's what we'll be referring to. So think about all the things you want to keep the same except for that independent variable. Moving on to the next thing, once we have that good setup and we have our variables, we want to collect some data. Before we start collecting just random data, you got to kind of think about, before the experiment starts, is what type of data are you going to collect? And so there's this quantitative versus qualitative approach. So those are our two. Quantitative data, I'll put a star by this and a smiley face, right? That's what we prefer in the scientific community most often. And most experiments that are good are going to lend themselves to some type of quantitative data, some numerical data to answer a question. So this data table over here shows quantitative data. We see the height of the seed in the Petri dish over time, and we can graph that and show a good change. The qualitative data is descriptive data, observational. There's no numbers, so like color change, observing a color change, that kind of thing. Both should be in a data table. Independent variable, when we graph in a spreadsheet, is column A. Dependent variables multiple variables if you need it, multiple columns, B through whatever you need. We'll do that in class. Units, every number should have a unit for the most part. There's some oddball exceptions, but common ones we'll be using for this class. Meter is the measure of length, gram is mass, volume is liter, time in seconds. Those will be the basic ones you'll get. And then we can slap prefixes in front of them. So meter is a pretty long unit measure, but maybe I just want to use the centimeter. So I slap centi in front of it, and a centimeter is 1/100th of a meter. We'll get, again, more practice with that in class, but those are the prefixes you'll need to know. Deci, centi, milli for small, deca, hecta, kilo for bigger units, typically kilometer or kilo is the one you see most often. Let's see, next slide we're going to discuss is our experimental design process and how it relates to something called our CER. The CER stands for claim, evidence, reasoning. Okay, so to start off, the scientific process you may have learned before is FIOC, problem, hypothesis, experiment, observation, conclusion. That's a pretty common way to present a scientific process or scientific methods. So we're going to kind of expand on that this year, talk about our problem. What are we trying to figure out? Well, that should be our research question. So that should be the beginning of the whole thing, our initial observation, our noticing. What are we trying to figure out? How does independent relate to dependent? The claim is really the hypothesis, and there's kind of two ways to think about it. The first way is it's your prediction. It's what you think is going to happen. But it should not just be like a yes, it's going to happen, no, it's not, so we don't want that. So it's helpful to structure it as a if, then, because. Claims can be revised after the experiment and saved for last to make sure that your claim is an accurate one that your data does support. Or you could think about your claim as a hypothesis that can be incorrect. It can be rejected, and that's okay too. Finally, after we have a good claim, we want to just design a procedure and then collect data. So our experiment is our procedure, and then our observations are really the data you collect. The data has to support your claim, support or deny your claim, but it has to relate to your claim in some way, and it has to be organized in a visual way. All right. So usually we talk about a graph or a table. Then we have a reasoning section. The reasoning section is really the conclusion. All right. So how do you know your hypothesis is supported or not? Use specific data. I'm telling you right now, you've got to have data in here. You've got to talk about your numbers and how they helped you form a conclusion of yes, this is supported, or no, it is not. So it's not quite as easy either as just listing all the numbers from your table and your conclusion. You have to use them to answer the question, and you really have to reason through that step by step by step. So we're going to look at a CER, a practice CER here. We will come back to this when we talk about scientific process and how this all applies in class. But just understand there's an initial observation. We come up with a question. We create a claim. We gather evidence and present it visually. And we have a reasoning section. So you could pause the video and go through this if you want to go step by step. Otherwise, we'll get back to this in class. So data. When we have our data, what do? Well, we have to graph. It always needs a title, y and x-axis variables. So understand we have the x is independent variable, y is dependent variable. Title's located here. We have appropriate scales and an easy to read graph. The scales need to be logical. It can't be just random numbers slapped on there. So even increments, logical starting point, that kind of thing. And labels should always have units on your axis. And we'll go over those in class some more too. Common types of graphing, scatter plot graphs, and then you may connect the lines or not connect the lines, and we'll kind of talk about how you determine that. But essentially, if we want to show a continuous change over time, we want to have that line present most of the time. So we're saying as depth changes, how does bubbles per minute there on the graph change? So the depth is continuous, and we want to see how depth affects bubbles. In the bar or column chart, you can call those either way, the same thing. You're comparing different groups to each other. So on the x-axis, we don't have continuous numbers anymore. We have names, groups of things. These are bacterial species here, and they're being compared to each other in creek versus forest. So it makes sense that we have a bar graph there. Finally, in your experiment, you do have to write and at least create procedures that can hopefully be followed by other students and myself. So generally speaking, we want to have procedures that do not write in the first person with I did this, we did that. You want to have your steps numbered. It's not some long big paragraph. And you want to include all steps and how you were able to record the data and what specific data you record, like what did you use to measure it, how often, like those really specific detailed things that we need to know. This is one of the more difficult things to do in a scientific process, and we'll definitely have some more practice with it in class.